Hello everyone! Good morning! Ayan, nandito na naman po tayo sa ating second day ng ating 26th Summer Institute in the Natural Sciences and Mathematics. Good morning po everyone! Ayan, so it's another day to learn about genetics and cell biology at the crossroads of different biological disciplines. Hello, good morning po everyone! Ayan, sana po maayos ang ating mga tulog at nakakain po tayo ng almusal. So, para po sa ating mga participants, kindly type in the chat box kung ano pong inagahan nyo kanina. Kung tinapay man yan or kape lang, pakilagay po sa aming chat box para naman po makita namin na ready po kayo for today's activity. Ayan. Talaga nga namang, ayan, meron tayong mga sagot ngayon sa ating chat box. Maraming Antaray, meron talagang may mga pa-leftover bulalo. Ayan, black coffee. Ayan. So, yesterday, mga participants and all of our SINSM Tech and Secretariat and facilitators, um, nag-discuss tayo about topics on cell biology. So, yesterday, we were introduced to different topics on cell biology at different biological disciplines by Professor Easter Joy Saho. So, yesterday, binigyan niya tayo ng brief lecture about, the different topics that we could do research on, particularly at the field of cell biology. And yesterday, nagkaroon tayo ng konting patikim pagdating sa demo on cell culture, particularly on plating, split, and freeze human cells. So, yesterday we were introduced to a new platform known as Labster. So, may mga different simulations and demo activities tayo na pwede nating ipagawa to our students. And in the afternoon, we also had a lecture on the different model systems. So, kahapon na-meet niyo yung mga model systems like the brine shrimp and all other organisms that we can use for bioassay techniques that we can also use for the research of our students and even the educators in our institutions. And I think one of the most insightful parts of our activity during the day one was our breakout session with the different biology professors in the department. So we had insightful discussions about conducting research during the pandemic and what are ways that we can do so that we could still do research and employ our learnings in the field of science. So I hope talaga na meron kayong mga nakuhang inspiration on your end so that you can still do research in your own institution. So again, welcome to day 2 of the biology discipline SINSM. Again, kumusta po tayong lahat? I hope hindi po ganun kainit this afternoon. So, hindi po kayo masyadong mas-stress. And we're really glad that you're here once again because we have another set of amazing speakers this morning and mamayang afternoon, and I hope na ready pa rin kayong makinig. I hope na ready na yung coffee nyo sa tabi ng mga tables nyo so that you're really, prepared to listen to amazing lectures and amazing demo this afternoon. So, without further ado, let me pass the mic to our SINSM moderator for this afternoon. We have here one of my colleagues in the department, Professor Aris Reginaldo. Sir Aris, take it away.
Good morning. So my task for this this morning session is to introduce our speaker, Dr. Madison Munar. We used to call him Mads, and probably because he is a young scientist that is also a graduate of the University of the Philippines, Baguio, and who took his master's and then just recently earned his PhD in molecular biotechnology. So we are very happy to have Mads or Dr. Madison or Munar this morning to share us with only a portion of what he has been learning in the past years. Parang basic ito pero yung kanyang mga research ay involved na parang hindi ko na rin maintindihan. My work is in ecology. I know a little bit also of molecular biology and biotechnology pero yung level ng focus ng recent work ni Mads ay parang napaka-detail na kasi I think these are marine or metal reducing bacteria. So let me now formally begin by formally introducing Madison. So Dr. Madison Munar, as I mentioned, has a PhD in molecular biotechnology. His recent work is focused on metal reducing bacteria. So if you want to Google about his work, so you can either and type his name or maybe try Googling ang tawag doon ay tellurite reducing bacteria. But in general, his recent work is focused on understanding how bacteria protect themselves or itself from the deleterious effect of the environment where they inhabit. Okay, so they have means to reduce or probably to convert the toxic material around them into something that they can tolerate. As I mentioned also, he's a graduate of the University of the Philippines. He's a young scientist. He used to work as an instructor at the department, and I think he's also one of our lecturers today, helping us with our teaching activities here at the university. He also worked at the molecular genetics laboratory at the Philippine Center in Nueva Ecija because I think he's also Esijano. Okay, so if we have participants here, probably you know Dr. Munar. So for today, Mads, he will be sharing to us one of the several things that he have been doing the past year. So for this morning, Dr. Munar will be presenting BLAST analysis. You've probably heard of this. This is an online open source bioinformatics platform. So allow me to welcome Dr. Madison Munar.
Good morning, sir.
Morning.
Good morning po. Should I start my presentation na? Yeah. To all my fellow educators, good morning. I'm Madison. Today I will be presenting my uh, lecture on online open bioinformatics platforms. So this morning I will be focusing on the first part, which is blast analysis, and this afternoon I will discuss another bioinformatics tool for phylogenetic analysis. So I'm going to turn off my camera so that you can view my uh, slides properly. Okay, so uh, this will be the outline of my presentation. I will start with a short introduction on the advantages of using DNA in establishing evolutionary relationships, followed by a brief overview on the methods employed in DNA sequence analysis. So, as I mentioned, I will be presenting the first part this morning, which is on blast analysis. And this will be followed by the second part of my lecture this afternoon on phylogenetic analysis. The objective of my presentation is to introduce online open source bioinformatics platforms commonly used in molecular phylogenetics. Mm. Nucleic acids are present in all domains of life, from prokaryotes to eukaryotes, including viruses. It carries genetic information and instruction that directs protein synthesis inside the cell. Nucleic acids are made up of the same building blocks called nucleotides, which include adenine, thymine, guanine, and cytosine. DNA sequence is the genetic material passed from parents to offspring or from parent cells to daughter cells. Thus, evolutionary relationship between organisms sharing similar characteristics can be traced by using this information. The nucleotide bases are essentially represented by only four characters: A, T, G, and C, which makes it very simple in terms of measuring or comparing the number of similar or different base composition in one DNA molecule between one species to another. Another advantage of using DNA sequence in reconstructing phylogenies is that it is consistent and not influenced by external factors which may arise from differences in the evaluation of phenotypic characters. In order to gain access or use the rich information encoded in the DNA is extracted from the organism under study and is sequenced. These techniques are becoming more inexpensive and can be performed in a very short period of time. The most important part in DNA sequence analysis is the assembly of the DNA sequence. After DNA sequencing, the DNA chromatogram, which shows the exact sequence of nucleotide bases in a DNA fragment, must be properly evaluated and has to undergo thorough proofreading. Short DNA fragments are manually assembled and should only include high-quality reads based on the chromatogram results. Normally, DNA sequencing companies will provide forward and reverse DNA sequences of the sample separately. These sequences should be quality trimmed in order to eliminate low-quality base reads prior to its assembly. Any single base change or erroneous base reads included in the final assembled DNA sequence can significantly alter the result of the downstream analysis, such as blast analysis and phylogenetic analysis. After DNA sequence proofreading and assembly, the final quality trimmed DNA sequence can be used for annotation using blast homology search and phylogenetic analysis. All DNA sequence data are stored in genetic sequence databanks that is easily and freely accessible for every researchers worldwide. GenBank, DNA Databank of Japan, and European Nucleotide Archive are the three DNA databanks where nucleotide and protein sequences are stored and all information are readily accessible through the internet. The three databanks are unified under the International Nucleotide Sequence Database. For instance, DNA sequence submitted and stored in GenBank is also accessible through DDBJ and EMBL websites. Now I will start my presentation on the first part, which is on blast analysis. This will include a brief introduction on the blast software and its primary use in DNA sequence analysis. This will be followed by a demonstration um, on its usage, and I will highlight important points in the interpretation of blast results. Okay. So basic local alignment search tool, or BLAST, is used to perform homology search between DNA and protein sequences. BLAST can rapidly match a query DNA or protein sequence from all reported sequences in DNA databanks. BLAST is a computer algorithm available for use online at the National Center for Biotechnology Information, or NCBI, website. This bioinformatics pipeline has connected biology, genetics, and computer science to further advance our knowledge in molecular analysis of DNA sequence and protein sequence. Um, there are 5 BLAST algorithms which require specific query data to be used in a specific database. BLAST-N is used for analyzing nucleotide sequences and uses nucleotide database. BLAST-P is used for analyzing protein sequences and uses protein database. BLAST-X, T-BLAST-N, and T-BLAST-X are variations of BLAST analysis which involves translation of nucleotide sequences and uses translated nucleotide database. These variations factors in the 6 spring translations in comparing translated nucleotide query. This is commonly used in the level of protein sequence homology search. BLAST software is very easy to use. It involves copying and pasting of the DNA or protein sequence or sequence identifier into the query text box, and the BLAST algorithm will perform the matching of every single characters in the query to all the reported sequences in the corresponding database. After which, BLAST results can be evaluated based on the coverage, quality of the match, and similarity of the query to the homologous sequences provided by the BLAST results. Okay, so for the demonstration, I will be using 3 common query data to demonstrate how to perform BLAST analysis. These include a DNA sequence. As you can see, it includes nucleotide bases, the A, T, G, and C. And we also have an accession number, a sequence identifier. So accession number is a newly reformed addition to the DNA databanks because it harmonizes the records of all the 3 data banks. And we're also going to use protein sequence with amino acid sequences in a single letter abbreviation. So I will start first in using the DNA sequence as my query. Okay, so uh, you can just type BLAST in the Google search engine, and uh, you can use it freely anytime using the internet. So you can see this uh, program where there is a text box. Uh, it says enter query sequence, where we are going to paste the uh, the nucleotide sequence that will be analyzed. Okay, so once pasted in this text box, so you can use the default settings. So it says database nucleotide collection and program selection, highly similar sequences. You can just click BLAST. So that's how simple it is. So once you click BLAST, it will then do the matching of the bases with all the other reported DNA sequences in the databanks. So this will be the result of the blast search. So if you're going to look at the result individually, uh, the first thing that you should look at is the query coverage, which is the percent uh, query sequence aligned to a sequence in GenBank or to a DNA data bank. And you should also have to look at the e-value or the expect value. So the lower the e-value, the higher the significance, meaning the alignment uh, is highly significant. So usually the first item on the list has the best score. So as you can see here, the first on the topmost list is Pseudomonas Stutzeri Hero Tree with 100% query, an e-value of 0.0, and a percent identity of 100%. Percent identity is the similarity of the sequence to the coverage. Okay. So after checking these three parameters, the query coverage, e-value, and percent identity, then you can have an idea of the identity of the DNA sequence that we are analyzing. So most probably, the DNA sequence that we use is from Pseudomonas stutzeri hero tree, based on this characters, having 100% query coverage, e-value of 0.0, and percent identity of 100%. So below this organism, it has lower percent identity, so lesser the match, lesser the similarity. Okay, so for example, there are really close match. Like for example, here you have uncultured bacterium with 100% coverage and 99.6% identity. You have to look for the uh, reference sequence, which is a complete genome of a particular bacteria in the same genus. So for instance, below that second organism, we have a complete genome of Pseudomonas stutzeri. So the genus and species is similar to the first one with the highest homology. So most probably, the first one is really a Pseudomonas stutzeri based on the presence of this reference sequence uh, on the blast result. Okay. So once you have decided that, okay, so it's Pseudomonas stuteri, so the topmost organism is probably the identity of the DNA sequence being analyzed, you have to check the accession number. So accession number, you can click on that accession number, it's underlined, and once you click on the accession number, it will bring you to the informations about the DNA sequence. So every uh, DNA sequence or protein sequence being submitted in a DNA data bank uh, are curated. Somehow they are peer reviewed. The informations, like for example, the authors, the source organism, the type of the molecule, the isolation are reported. So these informations are highly important when analyzing or using blast analysis. So you always have to look at the accession number. You just can click on it and uh, go through the informations provided. So another thing that you can do in blast analysis is there's FASTA on the top. Let's go back. There's FASTA below the name of the organism. So you can click on the FASTA and it will provide the uh, the sequence of the nucleotide or protein of the organism under study. So usually the FASTA format is used in phylogenetic analysis. So we are going to use this format later this afternoon. Okay, so the other variation that uses nucleotide sequence in the BLAST software is the BLASTX. So as I mentioned earlier, in BLASTX, the nucleotide sequences as query are first translated into amino acid, and then it uses a protein database to look for the homologous sequences. So it takes into consideration the 6 frames of translation of the amino acid. So let's see what will happen. So this nucleotide sequence is the same as what we have used earlier. So what will happen if we use BLASTX instead of BLASTN? Okay, so this will be the result. So again, we have to check the query coverage, the E value, and the percent identity. So uh, we can see here that the query coverage is 51, E value is very high, and the percent identity is also very low. So most probably, this is not the identity of the nucleotide base that we are looking for. So, so the higher the E value, the lower the significance. Okay, so if you can also check its identity, the informations about uh, the first one on the top list is hypothetical protein. So if you click on the accession number, so this is the information. So it shows the amino acid sequence instead of the nucleotide sequence, which we obtained using the BLASTN. Okay, so now we are going to use an accession number instead of a DNA sequence. So accession number, you can find accession number in all publications, in journals which deals with molecular DNA sequences or protein sequences. So accession number, you can use that in the 3 DNA data banks. So you can, if the sequence is reported in GenBank, you can use the accession number in the DNA data bank of Japan or in EMBL. So So they are unified. And the accession number harmonizes the record of all the DNA sequence reported. Okay, so if I wanted to study a specific gene, I'll just look for a journal dealing with that gene, and then, of course, once since it's already published, it already contains the uh, accession number. So I'll just take note of the accession number and use BLAST to to look for the information about the gene or protein maybe. So let's, for example, so going back to the BLAST software, BLAST site, you pasted the or input, you have inputted the accession number in the text box, and you click on the BLAST end. Okay, so uh, so it provided you a result. Shewonella algae hero 1 gene for 16S ribosomal RNA. So again, we have to look at the query coverage, the e-value, and percent identity. So here, it's quite high. So 100% query coverage, 0 e-value, and 100% identity. So most probably, this is the identity of that accession number that we have used or we are interested to study. And then, as we go below the list, there's still some reference sequences, complete genome, with the same genus, and species, Shewonella algae, still. There are there are other several complete genomes. I just captured a fraction of the result. Again, we all we have to check the accession number. So click on the accession number. Okay, so here, so the information about the identity of that accession number that we have used and the uh, complete DNA sequence. DNA sequence. So, for example, you are also interested in amplifying that gene. You can now use this sequence provided in BLAST to design your primers. So that's the advantage. So if you want to just get the exact sequences, click on the FASTA again, and it will give you the FASTA format of the DNA sequence. So you can use that sequence to design the primers and use the design primers to amplify the same gene for your study. Okay, so not all accession numbers can be used in BLAST-N. There are accession numbers that are submitted with a protein or amino acid record. So for example, this accession number that I use, I entered in the text box and then I use BLAST to analyze this accession number, and then it will give you a prompt. It says, failed to read BLAST query, GI, accession sequence mismatch, nucleotide input required, but protein provided. So it means that this accession number is not a nucleotide, it's not for nucleotide a database. So once you have that prompt, you can just change the your parameters into for to match the accession number into a appropriate data bank, which is for protein. So if you click on BLASTP, which deals with uh, protein data banks, then you will have the result. So using BLASTP, it says here the accession number that I provided with a protein record. It's probably a PAP2 superfamily protein. And we have to look again at the query coverage, the e-value, and percent identity. So it's very high. It's quite high. 100 coverage, 0 e-value, and 100% identity. So most probably this is the identity of that accession number. So we can also, again, click on the accession number and check for the information about the accession number. So, so it really has a protein sequence instead of a nucleotide sequence. So you can't really use BLASTn in analyzing the identity of that accession number in this case. But BLAST is very helpful because it will provide you a note that, okay, so you need to change the parameters. You should probably use another database for your uh, BLAST search. Okay, so now we are on the last a type of information that is used for BLAST analysis, which is a protein sequence. So the usual protein sequence is in the single letter abbreviation of amino acid, as you can see here. So I'm just going to copy this amino acid sequence and again paste in the text box in the BLAST search. So uh, if you click on BLASTn, again, it's not going to match with the proper database. So when using a protein sequence, you have to use a protein data bank. So instead of BLASTn, you have to use BLASTp. Okay, so just change your parameter to BLASTp, and then after changing, usually the default settings are used. So it's for BLASTp non-redundant protein sequences, and so for program selection, BLASTp protein to protein BLAST. So the protein sequence will be analyzed using the protein data banks in the protein data banks. So this is the result of the BLASTp. So there is only one homology based on that amino acid sequence that we use. So this again, this amino acid sequence resulted into one or single identity, which is a PAP2 superfamily protein. And again, we have to look at the query coverage, e-value, and percent identity. So as you can see, the query coverage and percent identity is below 60%, and the e-value is quite high. So again, so standard operating procedure, we have to check on the accession number to look for to look for the information about this protein. So okay, so these are the informations about the probable identity of the amino acid sequence that uh, we are interested in. Okay, so uh, but for example, uh, that amino acid sequence you obtain then from a publication which states that okay, so this sequence of amino acid is a protein coding gene. It is responsible for the production of this enzyme or this metabolite that can be used into a specific application. So in order to search or provide or get more information about protein coding genes, instead of using BLASTp, which is a more generalized homology search on protein databases, you can use the variation, which is a T-BLASTn. It takes into consideration the translation of amino acid sequences. So usually T-BLASTn is used to annotate protein coding sequences. So using the same amino acid sequence that we used earlier, now we are instead of using BLAST, I'm now going to use a T-BLASTn. So to investigate whether it is really a protein coding gene. So, so this is the result of the T-BLAST-N. So it says it's a bacterium related to telluride, a reduction. So again, looking at the query coverage, the e-value and percent identity compared to the result using BLAST, the query coverage here is much more higher. The e-value is much more smaller, and the percent identity is higher. And again, we have to look at the information about this uh, data by clicking on the accession number. Right. So here, these are the informations about the protein coding gene, and as you can see, uh, it was submitted with nucleotide sequence instead of amino acid sequence. So for me, if I want to study a protein and amplify that protein uh, in my organism, it's more helpful to get the nucleotide sequence where I can design my primers for PCR analysis. So, and so going back, since the parameters that we are looking for, higher query coverage, lower e-value, and higher person identity, is much more favorable in this data, then most probably uh, I will use this information instead of the one presented in the BLASP uh, result. Okay, so so after all our analysis, these are the BLAST annotation results. So using the DNA sequences, the query that we use is a nucleotide sequences, DNA bases. So annotation is most probably it is Pseudomonas stutzeri, strain hero 3. For the accession number query, so the most probable identity is Shawonella algae, strain hero 1. And for the protein sequence, as a query, so the most probable identity is the uncultured bacterium A1 DNA related to tellurite reduction and tellurium formation. So that's how simple, how to do BLAST analysis. Okay, so so how can I use BLAST analysis in my research? So if you're dealing with, if you are interested in discovering uh, proteins, enzymes uh, with industrial, pharmaceutical, or environmental applications, uh, uh, you can use BLAST as the first uh, method of analysis for your research. So this example is uh, more particular to bacterial cells because it's much more easier to culture bacteria and to collect bacterial cells. So if you're interested in discovering new enzymes or bioactive compounds from bacteria, you can just collect or first culture the bacteria, showing the activity that you're interested in, collect the bacterial cells in microcentrifuge tube, and you can store it in 40% to 50% glycerol. So instead of, uh, for example, the bacteria, the organism under study is still unknown, and you want to know its identity without going through the laborious process of primer design, DNA extraction, PCR, electrophoresis, and all. You can actually uh, send the bacterial bacterial cells stored in glycerol to a DNA sequencing facility. So, So you have to communicate with the DNA sequencing facility if they will accept that sample. But in molecular biology, bacterial cells can be used right away in PCR reactions, and then because in sequencing facility they have to amplify the DNA first before sequencing. So the DNA cells that you have collected can serve as the template. So it's called a colony PCR. So they will just use the cells directly into the PCR reaction, and then afterwards they can do the sequencing without DNA extraction. So there are fortunately we have now Philippine Genome Center that provides DNA sequencing services. So other DNA sequencing facilities that we have near the Philippines, in Singapore, there is a first base sequencing company, Solgen in Korea, or Eurofins in Japan. So, so you can choose whichever is comfortable. But of course, we we now have a sequencing company in the country, so why not utilize our own facility? So after if they accepted the bacterial cells that you have without extracting the DNA, then uh, better because you can save time and money in processing or identifying your sample. So so after getting the DNA sequence from the DNA sequencing facility, then so you have to do the proofreading and assembly of the DNA sequences. And after getting high quality DNA sequence, you can do blasts. So those blast results that you have is an information already that is publishable. So so that's a shortcut method in using blast analysis in research, especially now during the pandemic. So uh, the mobility and accessibility of laboratories is hampered. So you can resort to this methodology. Okay, so uh, uh, in the Philippine Genome Center, this is you can see this on their website. The cheapest pricing on DNA sequencing
Nuggets.
using Sanger sequencing, it costs only 250 pesos per sample. So I think that's very cheap. But um, per experience, not all sequence results uh, can be analyzed. So it depends on the quality of the sample. So mostly you really have to do the DNA extraction, the PCR part. But I have an experience that we only use the bacterial cells. We submitted that into to actually sa Solgen in Korea. We submitted our sample to Korea, and then we get good quality sequences. They have sequenced the 16S ribosomal RNA on our sample using the bacterial cells without going through the DNA extraction PCR on our part. Okay, so for the summary, so DNA sequence analysis using BLAST is very simple. Uh, you just always have to check on the accession number to get information about the data, and you also have to take into consideration the consistency of your query and the database that you're going to use. But the BLAST software can prompt you if you're using the right setting right away. Blast software is an open source, meaning it's free and it is user friendly. It's very easy to use. It's a copy paste method. So if you have a DNA sequence from a journal, you just can copy that, or an accession number, you just can copy that, or an amino acid sequence, you can just can copy that and paste on the blast text box and click on the blast search, and it will readily provide you the probable identity of your sample. So it is fast and easy. Usually it takes less than 5 minutes to get the result. And you can access the information stored in DNA databanks anywhere in the world. So So that's all for my for the first part of my lecture. So if you have any questions, maybe uh, uh, we can entertain it before we end this first part of the lecture.
Okay, thank you very much, Madison, for the very informative lecture. Mukhang madami tayong questions hope sa ating mga participants, and I hope for those who have joined us today, you take this time to ask Mads about all the inquiries that you have. Now, since he ended earlier than what is expected, maybe we can also use the time to entertain the questions. Now, Dane, are you there? Dane, or for the technical team? So how should we get the questions from the participants? So they can type on the chat box, or if you want to speak, I think our co-hosts can recognize you. So let's now open this time for the question and answer. Someone is raising his hand or hand. So can we Sir Antonio Lorenzo Ong, you can please kindly introduce yourself and your province, and then you may throw your question immediately.
Hello pa. So, yun, I am Antonio Renzo Ong, and I'm from Cainta Rizal. I would like to ask po, since it was earlier stated that in order to do a blast analysis, we need to do a parang genomic sequencing of the DNA first. So, I have some professors who said na most likely our thesis will be of a theoretical nature. So, parang it will be hard for us to do a genomic sequence in the lab for our thesis work since um, sabi ng prof ko na baka we won't have lab nga for our thesis. So, um, how do we use blast analysis po? We can't do for our thesis po, kung ganun po.
Okay. Uh, sorry, I didn't get your name again. What's your name?
Antonio Lorenzo po, pero my friends just call me Renzo.
Okay. Renzo. Uh, uh, kahit ako, even me, I wasn't able to do the whole genome sequencing. First, it's very expensive to do genome sequencing. So usually, for independent researchers like me, without any project yet, uh, 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 small DNA fragment sizes uh, is feasible for DNA sequencing. Or for example, in my example, in the shortcut version, where you can use the bacterial culture to be sent for DNA sequencing. The price is lower as compared to the whole genome sequencing, which could be like thousands, like more than 5,000 to 10,000 or even more. So usually if you're going to identify, for example, a bacterial species, they're going to amplify the universal uh, marker. For bacteria, it's 16S, a ribosomal RNA. So these are just short DNA fragments, about like siguro less than 2,000 base pairs. So it doesn't require much fund to sequence this kind of samples. So if you have maybe at least 5,000 pesos, you can have a five samples sequence for analysis already.
Odds. I think the other question, the other thing that Antonio wants to ask is very specific now, but I think this is related to thesis maybe. So I think he is asking also for a question particularly on what can you recommend to them, especially kung pwede nilang i-access yung blast, ganyan. So maybe what problem can be generated kasi given na hindi nga sila makapag field work or they cannot do laboratory. Is there any possible research proposal that can be generated or that can be created out of yung ni-lecture mo yan sa atin?
Yes, sir. Sir, yun nga po. That's why I added the last part where probable research topic using simple blast analysis. A shortcut method where they can just collect bacterial cells. This is more specific on dealing with bacterial cells because bacterial cells are much more easier to deal with. So, and in molecular biology, we use bacterial cells directly in PCR. So, without doing the DNA sequencing, the PCR, and all, we can just send these bacterial cell samples preserved in a glycerol stock to sequencing facilities, and then if they are capable or they will they can process your sample without providing DNA extracted DNA, then actually we have done that before. So the sequencing company in Korea sequence the 16S ribosomal RNA in the bacterial cells that we provided using that shortcut method that I presented without going through the process of culturing the bacteria, extracting the bacterial DNA, amplifying the amplifying 16S DNA using PCR, and then purifying the 16S rDNA amplified after PCR, and then eventually submitting that purified sample to the sequencing facility. So all those steps can be actually cut using the method that I have presented on the
Okay.
last.
So one more related question to you, to the question of Antonio.
Yeah.
So what if they also do not have bacterial cells? Can they use the information from the database to conduct a study or publish a research?
Okay, sir. Usually, if you don't really have access to the lab, or you can't do the collection of bacterial cells in silico analysis, so you can just do meta-analysis. For example, if you're interested in the phylogenetic or evolutionary story of a certain organism, bacteria, for example, or virus, for example, SARS-CoV-2, the for the SARS-CoV-2, the genome sequences of SARS-CoV-2, there is a designated data bank for the whole genome sequences of SARS-CoV-2. You can actually access that. You can use the accession numbers provided. And then, as I mentioned, the accession numbers can lead you to the information about the sequence and the actual sequence submitted to the data bank. So using those information, you can uh, uh, use the data for later. In the afternoon, I will be presenting phylogenetic analysis. So in the talk that I will be doing this afternoon, that's that's another uh, method that you can utilize using only DNA sequences from the web, from the DNA websites. Ay, sir, nakamute.
Sorry. Okay, so Antonio, so we hope na-answer namin yung question mo. If there are follow-up questions, so we can ask you again later on. But for now, we entertain other questions from our participants. I see a hand raised. Nawala. Any question po? Dami natin participants today. I think 100 plus. Okay. Anyone? So,
Hello, sir. Good morning po.
Good morning. Please introduce yourself.
Yes, I am Reggie Benigno from the Schools Division of Quirino, Region 2. Gusto ko lang po tanongin if there would be possibilities to have methods in extracting DNA of invasive alien species from fresh water like rivers.
Freshwater, come again? Freshwater.
Fresh water po, like rivers.
So parang any species from freshwater
Yes,
habitats.
yes, yes, sir.
Okay.
Okay,
Mads?
DNA extraction. So, although it's not part of my talk, there are readily available commercial kits for DNA extraction. For example, if you're going to deal with bacterial cells, for eukaryotic cells, for plant cells, there are specific DNA extraction kits for this already. But in general, the conventional method for DNA extraction that you can use is the CTAB method. Although this CTAB method is a bit toxic and requires a little training for processing. So usually in the lab, we use kits, but in certain cases, for example, if you're dealing with like environmental sample, these kits would not be effective. So instead of using kits, we use the conventional method, which is the CTAB method. So again, there are commercially available kits for specific type of sample for bacterial cell, for animal cell, and for plant cells.
Explain briefly yung CTAB method, Mads?
Okay, sir, actually, yung CTAB, madali lang siya. Although it uses chemical, so uh, uh, chloroform isoamyl alcohol. So you just add that into your sample, and then it will denature the cell wall of the organism under study, and then it will separate the the protein and the nucleic acid fraction. So usually the protein fraction are separated below the after centrifugation, and then the nucleic acid part can be extracted on the aqueous layer, which is usually on the top layer of the tube. So on that separation, on that manner of extraction, that's the conventional method. Although it's not highly purified because there could be some protein contaminants present in the DNA layer, but still you can perform several uh, successive extraction using the CTAB in order to get lesser protein contaminants on the aqueous layer which contains the DNA.
Okay. Thank you, Mads. Sir, nasagot po natin ba yung tanong nyo? Nasagot namin?
Yes, sir. Thank you so much for the information. Good day.
Okay, thank you. So we again ask for other questions from our participants. SINSM Secretariat is raising his hand.
Hello, Sir Alice. Good morning.
Morning.
For the benefit na rin po ng mga nasa YouTube live stream and those who can't turn on their audio, we have participants who ask their questions sa chat box. And to facilitate them, we have collated their questions and the tech team would flash them now. So, yun lang po, sir. Ako,
So I'm
paki-ask
okay.
nyo
I'm
lang
also
yung
seeing
questions.
several questions sa chat box natin. So maybe I'll read.
Sir, bali, we have tech team
Okay.
collating them. So, kahit yun na lang po yung basahin niyo, sir.
Okay.
Thank you.
Sige.
Ayan po.
So for anyone na gustong mag-speak din, so kindly just introduce your name, tapos you can ask the question while I'm looking at the other questions. Okay. Since wala pa ko ibang nakikitang question, but I'm able to see yung
Sir,
may question dito. Yep.
close your gallery view, sir, so you can see the screen
Sorry.
share.
Okay, sorry. Okay. But for now, may nakita kong question dito, Mads. Sabi niya, would it be possible to have the methods on how to extract DNA? Maybe they are asking kung pwede mong i-share yung methods on how to extract DNA. Meron ba pala tayong lecture on this?
Actually, sir, pwede siyang another topic for lecture. So, I wasn't able to include that since I only focus on the analysis part, bioinformatic DNA sequence analysis part. But to start with, all publications which dealt or reported DNA sequences, they included the method of DNA extraction in their paper. So that's the quickest way to get access on the method on how to extract the DNA from the organism of your interest. So, for example, if you want to know the method for extracting bacterial DNA, there are actually a lot, and there are several companies who already provided the methods, the step-by-step method using their kits. And if you are not interested in using kits, then you can actually use the CTEB method, which is also available in any publication which uses the method.
Okay. Pat, can you help me out in reading the questions na nakalate ninyo?
Okay, sir.
Okay, sir. Sige, go ahead.
So this question is from Mr. Christopher Cabado. Would it be possible to have the DNA methods on how to extract DNA?
Pat, we were just done with that. So next question.
Okay, sir. This one naman po is from John Raymond Torres. Good day, Professor. I am curious to know po about the colony PCR. Bacterial cells are used directly into this machine without DNA extraction. Maaari po ba na ang sample ay ipadala ay hindi po pure isolate? Like what are the species present in the culture from a swab?
Okay. Okay. Sir John Raymond. Okay, so colony PCR, ideally, the sample to be sent is pure culture, because if you're going to sequence mixed cultures, then the sequencing results will also be a mixture of different DNA sequences. So for that's the most important uh, factor in using colony PCR, using bacterial cells directly into the PCR. So you have to do purification of your bacterial species for colony PCR. But yeah, so if you're going to do the colony PCR, strict plating will do. So if you know strict plating, then you can purify your bacterial isolate. Then after purifying or getting the single isolated colony, you can reculture that and uh, collect the cells, then that will do for colony PCR. Or because you can if you have the PCR in your laboratory, and uh, you can actually use the colonies on the plate, and use those colonies directly in your PCR reaction. But in my example, I failed to mention that the collected bacterial cells should be pure culture, so should belong to one bacterial species in order to get the identity of that particular organism. In 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 case that you have a mixed culture, a non-purified culture of bacterial cell that you want to sample, uh, colony PCR is not the method of choice. So you cannot use the shortcut method. But for that sample dealing with mixed culture, different bacterial cells, environmental sample, you can use the CTAB method for DNA extraction.
Okay. So thank you. May mga ilan pa tayong questions dito from Pat. Can you read one of the questions from YouTube na meron tayo?
Okay, sir. Ayan. So from Setiram Sanchez, what an awesome blast, sir. Thanks for the great sharing po. Do you have sample titles of studies po? Yun po yung tanong.
Sample start. Maybe she's asking one of yours, your published paper.
Um,
Sample titles.
where you can, sorry, sample titles for a probable research topic or a publication.
Baka yung publication mo, banggitin mo na yung pinaka-recent mo. Marami, di ba?
Recent,
Maraming studies on this na.
sir, yung the DNA sequence, the accession number, and the amino acid sequence that I use in my examples were all from my publication. So you can actually get a copy of my PowerPoint presentation, and if you use the same DNA sequence, accession number, and amino acid sequence in my presentation, and do the blast search on your own, you can get the information. Just click on the accession number, and you can find my my name in the authors.
Yeah. Okay. Okay, we have another question from YouTube from Princess Sarah Quintana. This is her specific question, but I think you can answer it in a way that you want to. Can we also use BLAST for DNA barcoding of species? Or maybe she's asking, after they did barcoding, can they proceed to the next step, which is BLAST?
Actually, blast annotation is also part of DNA barcoding. So for DNA barcoding, you are interested in the identity of a particular organism using specific primers. So once you obtain the sequence from the organism under study, of course, BLAST is a must to annotate or get the identity of that organism.
Okay. So the tech team can share to us a screen share of questions. So if can you flash that so that we can read it, and or the participants can see the questions and then can ask them. Meron ba, tech team?
Ito po. From Mr. John Raymond Torres. Hello again, Professor.
Ayan.
I have another. So if there is only one homolog found on BLAST database, but the percent identity is low, how do we decide now on the identity of the amino acid we analyze?
Yan, pang-exam question yan, Mads.
Okay.
Sabi ko.
One homolog in class, percent low, and what is the type of query that you have used? DNA sequence depends on the query.
Maybe the protein sequence, Mads.
For example, the protein sequence is for example, the blast result provided single homolog based on the question, and there is a low query coverage. There is high e value, and then low percent identity. Most probably, the the protein is a novel protein. It's not
Okay.
there's no homologous sequence yet reported on the DNA database. So just what I use in the example, the uncultured bacterium. So the e value is quite high, but the coverage is uh, also high. So that the sample that I've used in my example is actually a novel gene.
Yeah. So, Mads, are you seeing the related question on the screen? Yung what if the sequence we analyze do not show any result? Will it be regarded as novel organism, or is it possible that it only happened since no data is uploaded in the blast system? Nasagot mo na ba yan?
Yes, sir. So similar with what I've mentioned, most probably there's still no data about the sequence you are dealing with. So in the example I use, it's a novel DNA sequence. Although there are similarity in DNA sequences, but this is not actually the exact identity of that particular nucleotide or amino acid sequence.
A related question to this, so kung ganun ba, pwede nang mag-identify ka ng new species? Ganun? Ganun ba yung ginagawa?
Sir, for bacterial cells, usually the gold standard is to use the 16S, so partial or complete 16 ribosomal RNA sequence to report that you have isolated or you have a new species or strain at least. You have to provide the complete or at least partial few base difference with the complete 16SR RDNA sequence for bacterial species. For fungi, you have the ITS, and for usually for prokaryotes, it's more complicated, so it's gene specific.
Okay, sige. Let's move on to the next set of questions. We have 5 minutes more. Maybe may iba pa tayong tanong. Okay, so this one from Baby College of Science, ito, how to target the right and the specific primers for sequencing. So I think this is outside of your scope, but maybe you can answer this.
How to target, right?
Or maybe identify the primers. Kasi parang yung kanina parang ang dali, parang kasi kanina yung premise parang ang dali na kasi, pero syempre may mga na-skip tayong method doon. And I think at the other aspect of indeed sequencing in doing the PCR is to be able to identify primers, especially for higher class of organisms.
Okay, so ay walang name, no? So how to identify specific primers for sequencing? So again, there are universal primers for specific organism that you can use, and these universal primers can identify similar organisms within the genus level. And then, how to target the right? Yeah.
Yeah, okay. So another question, can BLAST be used to detect and identify cryptic species?
If those cryptic species have records already in the DNA data banks, BLAST, DDBJ, or EMBL, then most probably it will provide homologous sequences from your BLAST inquiry.
Okay. Okay. So I think one more set. So if we have some more set of questions, do we have, Pat?
Yes, sir. I will flash it.
Is that a question? Okay. So by the way, ang dami na actually publications about all of the things that we are discussing now. So try to Google lang. Probably kasi pag may na-download kayong paper, medyo nakakatakot talaga kasi makita. But then since our scope is in discussing the methodology, take time to go to the methodology sections of those paper and you'll begin to see na parang pare-pareho lang din pala yung parang mga ginagawa dito. What we are doing now is more of the detailed aspect of that. So maybe you can ask more specific on those things, and even later on dun sa next part ng lecture ni Mad sa hapon, pag na-discuss niya yung phylogeny, baka mas doon ka ma-excite kasi ang daming research na pwedeng gawin. Yung aspect na ito, pwedeng gawin ng research assistant, mga ganon, assistant, pero yung iba parang analysis, iba matindi pang matindi pang trabaho yung pag-analyze, pag-interpret, and discussing all of this later on. So another question from Christopher Cabadu. Sabi niya, what if or maybe what is Drosophila, sir? May idea po ba about the squash method? It seems like this is an unrelated question, but maybe they're interested in knowing what Drosophila is and then the squash method. Maybe maybe the extraction ng chromosome siguro ang tinatanong dito, Mads.
So most
So you
probably,
can answer.
I'm not sure, but probably homogenization ng eukaryotic sample, Drosophila fruit fly is first a eukaryotic organism. So maybe they are interested on how to get the DNA out of this eukaryotic sample through the squash method. So most probably this is dealing with the homogenization, where
Na part pa lang.
you can still preserve the integrity of the DNA and use that for a downstream analysis such as DNA sequencing. So usually if
So parang kumbaga after the squash method, ito yung submit nila for PCR sequencing pa, no?
yes sir, so most probably it's on the DNA extraction part, and DNA extraction part is sensitive, the most sensitive part, because if you obtain degraded DNA, then all downstream analyses are affected.
Okay. One more, maybe one more. What we can submit to the DNA database or bank for analysis if I don't have the DNA sequence, or I just submit the whole organism and let them analyze or determine the DNA sequence. Baka ito yung nabanggit mo kanina na may mga laboratory na sila na yung gagawa ng sequencing. Tama ba? So may ganun option even for maybe eukaryotic animals or organisms.
Sir, if you want to submit DNA sequences in a DNA data bank, so it's somehow the submission is peer reviewed. So information, as I've shown in the accession number, once you click on the accession number, there are various information: the author, the sequence itself, the source of the DNA, the type of molecule, the product of the molecule should be provided upon submission. So the DNA data bank, they are going to check all this information prior to acceptance. So if they think that the sample is not doesn't have integrity yet in the first place, they will not accept or record that or save that in the DNA data bank. So submission in the DNA data bank is the first step in publishing paper dealing with DNA sequences. So it's not easy to submit and record any DNA sequences in data banks.
Okay, so we don't, I'm sorry everyone kasi wala na tayong enough time kasi ang dami nating participants, so we can only accommodate few. But maybe Mads, for the interest of for the participants who are just beginning to venture on this, we understand na marami tayong DepEd taga-DepEd na participants. Maybe briefly, no, yung summary lang ng sequence. Kung meron akong organism dito at gagamit ako ng molecular technique, ano yung simula nun at ano yung dulo nun? Kung gusto ko i-pursue yung research na ko. For example, meron akong species ng unknown species dito ng organism. Ano yung simula nun, tapos paano yung end?
Sir, yung pinakita ko kanina, so yung sa how to use DNA sequence in establishing phylogeny. So first, you have to extract the DNA. And then after extraction, uh, after DNA extraction, if you're interested in a particular DNA, probably a protein coding sequence, then you have to look for primers, reported primers on that particular gene, or if you have idea on the sequence, you can design your own primers. So you can use those primers in PCR to amplify that target DNA sequence. So after amplifying that DNA sequence, you can purify and then use that for sequencing. Then after sequencing, the sequencing company will provide you the actual DNA sequence, and then you can do the blast sequence homology search that we have talked today, and then or later the phylogenetic analysis for annotation and analysis.
Okay, so how about yung sa pag-publication na, ganun, for example, gusto mo i-publish yung work mo?
Sir, again, the first requirement to publish a DNA sequence or a paper dealing with molecular data, you have to submit the DNA sequence into a DNA data bank. So, for example, in NCBI, BLAST, in DDBJs in Japan, or in EMBL. So uh, uh, they can check your submission, and if all requirements are met, then they will provide you with the accession number that you can use in your publication.
Okay. Okay, so thank you very much, Mads. So give a clap. Let's clap. Virtually give our clap to Madison for a very good topic today. So again, we apologize everyone na hindi namin ma-entertain lahat ng questions nyo. But if you have more very important questions, you can ask him again later on sa topic nila sa hapon, so afternoon session. So we are formally ending our session this morning, but let me just announce daw na meron munang picture taking na magaganap. So I think you can turn on your camera with good lighting, ganyan, para makita kayo. Otherwise, I think it's best to use your photograph, ganyan. And another announcement, they're asking me to announce na yung evaluation, okay? Form, please don't forget to accomplish those. Okay, so technical team, you're ready for the picture taking.
Wait lang po. Oh,
100 participants ito, so hindi ko alam kung paano yung gagawin. Bahala na kayo.
On lang po ng ating mga cameras. Thank you. 1, 2, 3. Smile lang po. Keep smiling. Wait lang po. Konti pa. Smile lang kayo. So, smile lang po. 5 pages po ang ating gallery. So, ayun po. Okay na po. Thank you. Maraming salamat.
Okay, so thank you very much everyone for joining us this morning session. So see you again this afternoon. Tuloy-tuloy po yung pagkatuto natin kahit online tayo. We know it's very hard for all of us, but we appreciate that you join us today and I hope you learned something from this session. So from my end, I'm Professor Aris Reginaldo. So thank you for joining.
So good afternoon, dear participants. Okay, I am Ron from the Department of Biology of the UP Baguio, and I will be your host and moderator for the Q&A for this afternoon session. Okay, so kumusta po tayong lahat? Kumusta po ang ating lunch? And I hope na just like us, you also had a great ano po? So I've heard na we have participants from Bicol. So si mga tugang ta dyan sa Bicol, kumusta man po kamo dyan? Nagkaraka naman po kamo ano? Abahan ng siram ka mga panira dyan sa Bicol ta. Namimiss ko na talaga. Si mga Bisaya man po, sa mga igsuon nato diha sa Visayas o sa pila ka parte sa Mindanao, kumusta man mo? Okay, murag na ay bagyo karon. So mag-amping po kita tanan. O sa mga Hiligaynon, ato ang mga kamarangho dida sa Iloilo o sa ila ka parte sa Visayas, hay maayo pa mo kay naamoy kanang mga beach diha. Kami diri sa Baguio, okay man, nya maano na, mainit-init na ang panahon. Yan. So, I guess okay naman po tayong lahat. Ano po? So, just a few reminders lang po. Kindly mute your microphones and turn off your camera, okay, during the whole session. And kindly field your questions on the chat feature of the YouTube live stream and chat box in our Zoom platform after the lecture po. Okay. So just a recap also, earlier today, Professor Munar discussed about the BLAST or the Basic Local Alignment Search Tool. And we were so lucky ano po, kasi we have learned about its concept, especially magagamit natin siya in identifying organisms at the molecular level. So for this afternoon, Professor Munar will also give us another lecture on molecular phylogenetics. So I hope excited din kayo katulad namin. So without further ado, let me call in Dr. Madison Munar for his lecture.
Share. Again, good afternoon. So this is the continuation of my presentation. So I'm going to close my video so that you can see my the whole slides of my presentation. Okay. So the second part will deal with phylogenetic analysis. I will provide a short introduction on molecular phylogenetic analysis, its application. This will be followed by a brief demonstration on how to use the MegaX software for phylogenetic tree construction. I will highlight important points on how to interpret a cladogram. Molecular phylogenetic analysis is used to infer evolutionary relationships based on nucleotide or amino acid sequences. Samples or data are grouped based on the similarities and differences of the genetic sequence. Phylogenetic analysis is dependent on the quality of alignment of the underlying data. Phylogenetic analysis is used in classification and identification of new species. It can aid in assessing DNA sequence evidence or in paternity testing. It is used to identify pathogens and source of transmission. It can also be used to discover novel bioactive compounds with industrial, environmental, and pharmaceutical applications. Moreover, it can help inform conservation policy about which species are about to become extinct. Now, I will introduce an open source bioinformatics software that can be used for molecular phylogenetic analysis. MegaX, or Molecular Evolutionary Genetic Analysis, is an integrated bioinformatics pipeline used for multiple sequence alignment of DNA or protein data to infer evolutionary relationships through phylogenetic analysis. This software can be easily downloaded from the internet. There are 6 basic steps in phylogenetic tree construction. These include exporting FASTA sequences, aligning exported sequences, inspecting the alignment, quality trimming of unaligned sequences, saving the aligned sequence, and finally, building of the cladogram. For the sample analysis, I will use 3 different strains from 3 bacterial species, and I will include a different strain as an outgroup. So the 3 bacterial species that I will be using are Shawonella algae, Pseudomonas oleoverans, and Pseudomonas stutzeri. For the outgroup, a different species is the Escherichia coli, strain U5 over 41. So I will be using the accession numbers of these reported bacterial strains in NCBI for the sample analysis. Okay. So for the first step, to export FASTA sequences into the MegaX software, click on the Align button here and edit Build Alignment. Then create a new alignment and then choose the type of molecule. In this case, we are dealing with DNA sequences. So click DNA. In order to add new sequences using the accession numbers of the organism under study, click Web and query GenBank. So this will bring you to the nucleotide database in NCBI website. Paste the accession number and click Search. You will get to the GenBank database where the sequence in FASTA is recorded. Just click on the Add to Alignment button on the uppermost part of the screen here. Okay. So there is a prompt that where you can add the labels of your DNA sequence sample. So just choose on the suggested label, for example here, Shawonella algae, the strain, and include the accession number. So after setting the sequence label, just click OK. Okay. So after, just repeat the process. Click on the web, uh, 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 query GenBank and paste the accession number and then add alignment. So after adding all the sequences for analysis, you can edit the labels of your samples. I highly suggest that you put the accession numbers in parentheses. So these labels will be reflected as it is in the phylogenetic in the cladiogram after phylogenetic analysis. So the second step is to align all the exported sequences. To do this, click any base and use the Control A to select all the bases. After choosing or highlighting all the bases, click on the Align the muscle program and Align DNA and use the default settings. So click OK and it will start to align the data. Okay. So the third step is to visually inspect the aligned sequences. So you can go from one end to other end to visually inspect all the alignment. And then as you can see here, both ends of the uh, multiple aligned sequences has sequences that are not aligned completely here. So these bases should be removed prior to the construction of the phylogenetic tree. Because those sequences that are not aligned properly or will be interpreted as polymorphic regions. Thus, similar species will tend to be grouped separately. Unlike if you are going to delete all those unaligned sequences and you're going to use sequences with exactly the same length, then the actual analysis on the different DNA sequence will be used. Okay, so to trim the excess sequence, highlight those unaligned excess bases and use the scissor icon or the X icon to delete. Okay, so the fifth step is to save the alignment. So to do this, click Data, Save Session, and then save the alignment in mega format. So just type the name of the file or alignment and click Save. Right. So after saving the alignment, we can now do the sixth step or the final step, which is the building of the cladogram. So to do this, just click on phylogeny on this icon, and then you can choose any building method. So for this example, I'm going to use construct test maximum likelihood tree. Then we are just going to open the save alignment in mega format. Okay. So here you have to set the parameters. I would suggest to include a bootstrap method here with 1,000 replications and a distance correction method like Kimura 2 parameter to ensure the quality of the phylogenetic analysis. So after setting these parameters, the bootstrapping and distance correction method, you can just click OK. Okay. So the amount of time of analysis depends on the length of the sequences being analyzed and the number of sequence included. So after analysis, you can save the image in PDF format. So PDF file can be used in publication because you can just copy the saved PDF image directly to the Word format without diminishing the quality of the figure. Okay, so this is the result of our example. So here you can see that all the Pseudomonas strains are grouped into one cluster, as well as the other Pseudomonas strains, Pseudomonas strains, are grouped into one clade. This is also true in the another species that we use for Shewanella algae. So the Shewanella algae strains are also grouped in a single clade. So there are three different clade, monophyletic clade uh, from our sample analysis, and you can see that our outgroup, the different species, which is Escherichia coli, is not included in any of the clades. So the the cladiogram can be represented in various three representations. For example, you can have it in the rectangular form, radial, or circular form. The most common representation used for publication is the rectangular tree. Okay. So in interpreting the cladogram, you have to look at the bootstrap value. These values are indicated at the nodes of the phylogenetic tree of the cladogram. So in our sample analysis, we have 100% bootstrap value in the group of Pseudomonas species, and also 100% bootstrap value for the clade of Shewanella algae. So bootstrap values above 90% is strongly supported, while bootstrap values below 50% is not supported. So in the clade of Pseudomonas oleoverans, the bootstrap values still above 85%. This is also true with the Pseudomonas stutzeri and Shewanella algae. There is a very minimum sequence divergence. Okay, so the sequence divergence is estimated by the scale bar uh, in the cladogram. So this scale bar indicates the number of substitution per site. So this length, so if you're going to compare this length to this length, so this length means that there are two base difference for every 100 bases. So the longer the distance, the higher the sequence divergence. So for those strains in the same species, you can see that they're very short sequence divergence because they are actually the same species. This is also true with this clade and this clade. However, as you compare this genus, Shewanella algae, with these two Pseudomonas species, there is huge sequence divergence. So it indicates that they are different on different genus. So this is also true with our outgroup. So it's really not included within the clades. All right, so I think it's quite fast. So, so I have presented this earlier in the blast analysis on how to employ these techniques. So for the blast analysis, we you can just collect the cells at the shortcut version, send these bacterial cells for sequencing, and do simple blast, and you can actually produce a paper, a scientific publication with that information. So, having the sequence and can actually be used for phylogenetic analysis. You can also extend your analysis further to get more or extract more information out of that DNA sequence using phylogenetic analysis. So um, for the summary, uh, molecular phylogenetics is used to elucidate evolutionary relationships, especially among prokaryotic organisms, including viruses. Phylogenetic analysis has become an invaluable tool in the discovery of new species or strains and in epidemiological studies. So, thank you for your attention.
And so thank you so much, Professor Madison, for that brief yet informative lecture presentation. So I would like to personally commend that lecture presentation on the basis that this is really something that we need to understand, especially in this time of pandemic, wherein different strains of SARS-CoV are being discovered. So the technique presented by Dr. Munar is really a relevant and timely practical application of science in today's world, especially that we need to trace these fast evolving strains, right? Okay, so we shall now go to the question and answer portion. Again, I would like to request everyone to please field your questions on the comment boxes in our YouTube live streaming channel or send directly your questions through our Zoom virtual platform chat box. Okay, so Secretariat, do you already have the questions, please? So while waiting for the questions, sir, I have a personal question. So the Mega po, what else na application or software can we use to easily do this po? For example, may mga students po na who want to conduct researches like this po. So ano po kaya yung ibang application that's open source na pwede nilang ma-access easily?
Personally, I'm only particular with the MegaX software, but there are several other phylogenetic tree tools. So I'm biased because I've been using Mega software since then. So, and since it's free, I'm not quite sure about the other phylogenetic softwares, but there are a lot.
Thank you so much, sir. So we have a question po. So the first question is from Miss or Mr. Jarel Casimiro. So first question is, can you give some tips in choosing an outgroup for the cladogram, sir? And the second question is, are there any considerations when choosing clustering method and distance correction method po?
Okay. I think si Jeriel is my student before. Hi, Jeriel. So tips in choosing an outgroup. The outgroup is not essentially required to be included in the analysis, but using an outgroup can uh, provide more integrity on the analysis. So since most phylogenetic analysis is based is unrooted, there is no sense of evolutionary. There is no root. Since we don't know the exact origin of the species, putting an outgroup actually uh, provide that sense of root, but it's actually not a root, just an outgroup. And then for the outgroup, usually we can choose a different species other than the species we are analyzing. So, for example, in my in the analysis that I did, so Escherichia coli is not does not belong to any of the bacterial species that I have chosen. So you can clearly see that it separates among the three clades uh, in the final tree. It's important to note that the outgroup, if it affects the alignment, because the cladogram depends on the quality of the alignment. So if the chosen outgroup is affecting the alignment of your sequences, then it's better not to add an outgroup. It's acceptable. So journals accept cladogram without an outgroup. And are there any questions? Okay, so there is no uh, particular uh, method that is advised to be used regularly by researchers. So every researchers can choose any building method. So if you're going to publish your tree, usually the reviewer will ask for at least three different building methods used in analyzing your DNA sequence or protein sequences in order to make sure that the the tree is based purely on the alignment and is not biased on the type of building method used. Yeah. So if you're going to include cladogram in your paper, make sure that you have also tried, because it's also simple. Just after trying uh, the maximum likelihood tree, then try the other tree building methods like neighbor joining, minimum evolution, UPGMA. So to be ready for the reviewers' requests for those data later on.
Okay, so now, thank you so much, Jarel and Sir. Um, we have another question here. The question is, are these software free, sir?
Ah, ay, Ma'am Marites, opo. So actually, yung Mega Expo is, uh, there are several versions already. So yung version that I've shown is version 10. So now there is version 11. So you can also use that. You can try that. The how to download Mega X is quite simple. Just type Mega software or the complete name molecular evolutionary genetics analysis in any search engine, and then it will bring you to that software. And the installation is also very easy. It's free.
So much for that question, Miss Maritas. The question is from from Mr. Jose Rolando Caingay. So yes, can we have a copy or any suggestion of a literature wherein we can dive in as well as the software? Yeah.
So far, when I started using bioinformatics tool, there is very minimal resources dealing with the discussion on how to use these programs. But now I've seen a lot of courses and books available online, which also discusses on the use of these bioinformatics softwares. Can have a copy, literature. Uh, Actually, Mega X software, they it is they have a publication, every bioinformatics tools or the concept, the methodology, how to use, these are all published. Most especially, for example, in high impact factor journals like Science and Nature. So you can see these bioinformatics tools published in those type of journals.
Thank you, Sir Rolando, for that question. Next question is from here's a follow-up question from Sir Rolando. So what are the criteria in choosing an out group? So do we really choose an out group, or it is an outcome of alignment?
Okay. Sir Jose, the out group should be a different species from the species that we are concerned, but it's not necessary to include an out group. If we try putting a different species and it affected the alignment, this will construe the information that will be produced after phylogenetic analysis. So, as I said, you can add alignment. It does not affect the alignment because the tree construction highly depends on the quality of the alignment. If it does not affect, then you can include that in the final tree. But if it affects the alignment of the analysis of your data, then it's better not to include an outgroup.
Okay, so next question, please. From Sir Kenneth Frias. Is it the new standard in taxonomic classification?
I think yes, because if you're going to report a new species, some journals will require a molecular DNA analysis at least. So for bacteria, at least the complete sequence of 16S RNA, for fungi, the ITS, and so on. So nowadays, molecular analysis for the description of new species is a requirement.
Next question. Anyway, so while waiting for the next questions. So sir, i-clarify ko lang po. So, at kailangan po ba talaga nasa molecular level tayo when it comes to classifying organisms, no? Or masasabi ko masyado kapag morphological basis lang tayo?
Since I'm working with prokaryote as a bacteria, so it's highly recommended to provide molecular data. But I think in eukaryotic organism for plants, morphological characters are accepted to report a new species. But molecular data can strengthen the publication.
I agree with you, sir. So here's another question from Ms. Minette Iloreta. So can we still use sequences already studied and use mega sort of meta-analysis or in silico study?
For in silico study using bioinformatics software, you can use sequences reported in the DNA data banks. So if you don't have your own sequence, you can do the meta-analysis research. If you want to prove that, okay, the organism really belongs to this clade or group, then you can do so using the phylogenetic analysis. It will support your hypothesis, or it may not support the hypothesis, but you can do so.
Thank you so much, Ms. Minette. I hope we answered your question. The next question is from sorry, medyo naglo-load lang po. Here's another question. So another question from Mr. Kenneth Barrientos. So how can we identify novel species using phylogenetic analysis? Thanks.
Okay, so Sir Kenneth, for example, uh, the you have a sequence and you reported that or submitted that in a DNA data bank. So you have an accession number. So using BLAST analysis, you can have the idea whether your organism is already a new species or a new strain on the BLAST level. So it will provide you the idea whether so it could be a new species or a new strain. So to further strengthen the claim of a new species, you can do the phylogenetic analysis. So in this case, if you want to really support the hypothesis that you have a new species, you have to include all possible species related with homologous to the sequence present in the DNA data banks. So you get them, record their accession number, use their accession numbers, and do the alignment. And include your organism that you want to report as a new species. So once you got the picture of the phylogenetic tree showing all the related organism, if your sequence that you are hypothesizing to be a new species showed at least 95 or 85 sequence divergence, the bootstrap value, it could be a new strain. So just a new strain. The species level, to report a new species level, you need to use at least the whole genome. For example, in bacteria, so you cannot report a new species by just merely using the 16S ribosomal RNA sequence. It's very short. It does not conclude that whether the organism is a new species or not. Just a new strain, probably. But for a new species, it requires a larger sequence, whole genome, at least.
Thank you, Sir Kenneth. Our next question is there a standard threshold value for species to be considered divergent?
Uh, uh, if you're going to look at the confidence value based on the bootstrap value, similar species uh, would have at least above 80% confidence value on their grouping. So if the confidence value or bootstrap value is below 50 and you can see that there is a particular grouping, then it's not accepted. So there might be some errors in the sequence or in uh, the the samples. You can check your samples, the data of the samples. You can go back to the record in the DNA data bank and review whether it has the same type of molecule or it has similar uh, the size, especially the size of the sequences, is very important in phylogenetic analysis.
So we hope it answered your question, Sir Kenneth. Next question is, I guess there's a comment, Muna. So it says here, congrats, sir, for the interesting talk. So the question is, will you also provide a talk on sequence cleanup and config assembly?
Oh my God. Actually, I have a lecture on that topic in my Bio 150 class. So why not?
Are there any other questions? So I guess that question is from Sir Patrick, okay, one of our colleagues here. So I'll just check the chat box. So, sir, may question dito sa Zoom chat box natin from Miss Charmaine Bactan. How important is it to start? How important is the study of phylogenetics in forensic investigations? Will it somehow provide a valid result po?
Usually, in forensics, you have to sequence the DNA material collected from, for example, in a crime scene. So uh, uh, they usually compare the genetic material from the crime scene to the suspects. So by sequence alignment, you can actually using electrophoresis, you can already see whether the sample belongs to this suspect or not. The phylogenetic analysis only confirms based on the actual sequence of the material.
So another question also, sir, from one of our colleagues. Okay, would the methodology be the same for all taxa, for animals, plants, fungi, and bacteria?
The methods that I've shown in exporting the FASTA sequence in the software, the alignment, the edit, the, the the cutting of the excess and unaligned sequences, the multiple sequence alignment, and the choosing of the tree building method is the same. Whether you're working with bacteria, fungi, or eukaryotic samples.
So yes, thank you so much, sir. So another question from Sir Jose Rolando Kainkay. Sir, can we also use phylogenetic analysis in contrasting two types of viruses?
Sir Jose, yes po. So there are a lot of publications right now about SARS-CoV-2, whether the 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 causative agent, the etiologic agent of SARS COVID-19 is a new strain or yeah, or a novel strain. So based on the phylogenetic analysis, for me, my interpretation is that SARS-CoV-2 is a new strain. Though it's not novel because novel strain is new, uh, entirely uh, entirely different or very novel. However, the SARS-CoV-2 is just a strain, a new strain.
So sir, while waiting for the other questions, I would like to read the comments in our chat box. So from Mr. Dane Frawley, so fan fact lang po for everyone, Dr. Munar was my professor in Bio 120 or microbiology and Bio 197 special topics, Introduction to Genomics and Bioinformatics. Super organized po ni Sir magturo. And then another comment here from Miss Mary Jane Bulusan. Thank you very much po. So informative. Okay. Aside from that, sir, okay, we have another question from Sir Kenneth Rias. So does molecular level need to coincide with morphological feature to strengthen phylogenetic analysis?
Yes. Actually, that's the most recent proposal that you have to combine the molecular data and the phenotypic data. So that's the most ideal analysis for evolutionary relationship. Yeah, I forget the term, but yeah. The molecular data should coincide with the phenotypic data.
So thank you so much, Sir Madison, for answering that question. So I think we have a couple of questions sa YouTube streaming natin. Okay. Or may mga magsasend pa ba ng questions through our Zoom virtual platform? Oh, so here's a comment. So ako rin po, student ni Sir Mads Munar, proud mentee here. Pasample naman po ng Korean. So it's from Mr. Geno Pagaduan. Questions? Okay, so I guess may mahap. Okay, another. Oh, here's a question from Mr. or Miss Razon. Phylogenetic analysis can be used in crime investigations, and having a gene bank can ensure that the authorities have copies of those DNA of criminals around the globe. So it's just a comment lang po pala. We have questions pa po. Okay. Mahaba pa po yung oras natin, so pwede pa po kayong magtanong kay Sir Madison. So I guess wala na pong questions, sir. So any final words po from you, Sir Madison?
In case po na you need help in using these techniques that I've discussed, this SINSM activity, you can reach me through my email or Facebook, and I'm very much willing to help.
So in case you have questions pa din po kay Sir Madison, you can reach him through his Facebook nga po and or dun sa page po namin sa Facebook para we can relay the question na lang, Sir Madison. So again, this concludes our lecture presentation for today. Thank you to all our participants and of course to our lecturers. So hope to see you in our next SINSM lecture tomorrow. Again, this is Ron from the Department of Biology. Have a good day.
And
we would also like to encourage everyone to please fill out our evaluation form or attendance form using this link shown on the slide. So this link will help us to better improve our SINSM lecture series. So thank you. Have a good day.
yeah, so thank you very much to all of our participants. And so don't forget to fill out the evaluation form for day 2. And yeah, that concludes our day 2. So please, hopefully, we will see you tomorrow on our last talk. We'll have day 3. So yeah, and again, thank you very much and see you all tomorrow. You may now leave the meeting. Thank you.